Summary.
Prologue. Families. Mukherjee's uncles Rajesh and Yagu and his cousin Moni have all suffered from mental illnesses with genetic origins. Their stories serve to introduce the gene, the fundamental unit of heredity that holds the key to the physical and emotional characteristics of each human life. Need to know: no understanding of human biology, physiology or behavior can be complete without first understanding the nature of the gene.
Part 1. The Missing Science of Heredity.
The Discovery and Rediscovery of Genes, 1865 to 1935.
The Walled Garden.
The concept of the gene was first formed by Gregor Johann Mendel in 1865. However, theories of how human traits are transmitted date back to Aristotle, who theorized that they were contained in instructions within the body that were passed on from generation to generation. Aristotle's theories of heredity were surprisingly accurate, but after him, little progress was made in genetics until the 19th century.
The Mystery of Mysteries.
Charles Darwin's 1831 to 1836 voyage to South America and the Pacific to collect animal specimens and fossils of their predecessors led him to believe that new species were created in a struggle for survival. The ones best equipped to adapt to shifting conditions would endure, in a process of natural selection. Need to know: Darwin's On the Origin of Species proposed the theory of evolution but did not provide an explanation for how heredity works.
The Very Wide Blank. Darwin later sought to explain heredity with the theory of pangenesis.
He believed that tiny particles called gemmules, contained within the cells of living organisms, passed on their essential traits to their offspring, blending the characteristics of both mother and father.
His colleagues dismissed the theory because it did not explain why some traits disappeared and then reappeared in later generations.
Need to know: Darwin's failure to explain heredity came about in part because he was a brilliant naturalist, but not adept at carrying out experiments.
Flowers he loved.
Gregor Mendel's research breeding hybrid pea plants led him to conclude that the traits of organisms must be contained in single, indivisible units. Although he did not invent the term, Mendel had identified the fundamental qualities of what we call a gene today. Need to know: Mendel's seminal work on heredity was received with silence. His contribution to science was not recognized until long after his death in 1884.
A certain Mendel.
The Dutch botanist and geneticist Hugo de Vries, aware of Mendel's work, observed that plant species spontaneously generated new varieties, ones with longer stems or flowers of different colors.
He termed these variants mutants, and the process, mutation.
Nature, he posited, spontaneously created mutants with specific qualities, and the ones best adapted to changing circumstances were able to survive, creating new species. Need to know: Hugo de Vries's discovery of spontaneous mutation extended Darwin's theory of natural selection. Eugenics. Darwin's cousin Francis Galton was an early proponent of eugenics, which advocated the selective breeding of men and women in order to create a superior society. The notion appealed to a British ruling class appalled over the rising power and numbers of the working class, and to sectors in the United States concerned about the effects of massive immigration. Need to know: the eugenics movement in the United States and Britain used theories of genetics to justify social experiments that included forced sterilization.
3 generations of imbeciles is enough.
During the 1920s, individual states began to pass laws authorizing imprisonment or sterilization of men and women deemed to have genetic criminal tendencies or mental deficiencies. The first legally forced sterilization was performed on Carrie Buck, an impoverished single mother in rural Virginia who, like her own mother, was confined to a penal colony after being diagnosed as a moron. Buck, who did not express any objection to the procedure, was sterilized under a 1924 Virginia law that was later upheld by the U.S. Supreme Court, opening the way for thousands of such forced sterilizations. Need to know: in the United States, eugenics became, if not the law of the land, a powerful tool for people who sought to protect society from what they regarded as moral and social decay.
Part 2. In the sum of the parts, there are only the parts.
Deciphering the Mechanism of Inheritance, 1930 to 1970.
Av.
Thomas Morgan, a researcher at Columbia University in New York, conducted extensive experiments on fruit flies and found that genes were linked to one another and to chromosomes. Based upon this notion of linkage, a student of Morgan's, Alfred Sturtevant, constructed the first genetic map. Need to know: Thomas Morgan's graphic description of genes' location on chromosomes as beads on a string illustrated their linkage to one another, and to chromosomes.
This was the first genetic map.
Truths and reconciliations. Theodosius Dobzhansky, a Ukrainian biologist who immigrated to the United States, discovered that while genes, or genotype, determine traits, phenotype, environment also has an effect, as does chance. He exposed groups of fruit flies to different temperature levels over several generations. The resulting changes in their physical traits showed that environment also influences an organism's genetic makeup. In this way, Dobzhansky also proved that gene selection alone could not be the basis for creating superior beings, as the eugenicists believed. Need to know, Dobzhansky's experiments led to the equation genotype plus environment plus triggers plus chance as the explanation for physical attributes, phenotype, of living creatures.
Transformation.
Frederick Griffith, a British medical officer seeking a vaccination for the Spanish flu, found that two different varieties of bacteria could exchange genes, with one acquiring the attributes of the other.
This meant attributes could be transmitted without reproduction, in a process he called transformation.
Hermann Muller proved that genes could be altered by radiation to produce mutant varieties.
This meant that genes were actual, physical matter that could be manipulated by man. Need to know, the research by Griffith and Muller marked the first step toward manipulation of human genes. Lebensunwertes Leben, lives unworthy of living.
The Nazis' mass experiment in racial hygiene began with sterilization and quickly moved to the imprisonment and execution of mental defectives, criminals, dissidents, Jews, and gypsies. The Russian communists, meanwhile, used theories of gene manipulation to justify a massive program to erase all differences in thought through brainwashing or re-education. Need to know, both the Nazis in Germany and the communists in the Soviet Union used the language of genetics, although not exactly the science itself, to justify their atrocities.
That stupid molecule.
In 1944, Oswald Avery, a professor at Rockefeller University in New York, experimenting with virulent and non-virulent bacteria, proved that the nucleic acid DNA, deoxyribonucleic acid, is the molecule that carries an organism's genetic information.
Avery based his discovery on the work done previously by Frederick Griffith.
Need to know, the identification of DNA, scorned as that stupid molecule by one scientist, as the carrier of the genetic code served as a landmark to direct future research in genetics. Important biological objects come in pairs. Researcher Rosalind Franklin, using an X-ray machine, took the first useful photos of DNA molecules, which permitted her colleagues James Watson and Francis Crick to determine the molecule's structure. After many attempts, they created the iconic double helix model that illustrates the nature of DNA, the precarious assemblage of molecules which holds the secret of life. Need to know, Watson and Crick's discovery of the structure of DNA in 1953 posed a new set of questions about how to manipulate that chemical structure to the benefit of humanity. That damned, elusive pimpernel. Knowing the structure of DNA, scientists sought to find out how the molecular model actually transmitted characteristics. George Beadle and Edward Tatum proved that the action of a gene is to encode information to construct a protein, which in turn enables the specific form or function, the trait, within an organism. Later, Watson and Crick discovered their central dogma, that RNA, ribonucleic acid, DNA's molecular cousin, is used as a messenger molecule to transmit copies of the DNA chain to build a new protein. Need to know, the central dogma of biological information is that DNA is transformed into RNA, a messenger molecule that constructs a new protein.
Regulation, replication, recombination.
Experiments by the French biologist Jacques Monod proved that genes were not just passive carriers of traits, but were capable of changing the information they held to respond to changes in the environment. He did so by exposing the bacteria E. coli, Escherichia coli, to two sugars, glucose and lactose, and noting the genes' different reactions to each one. Monod showed that genes are able to regulate themselves, to replicate themselves, and also to recombine, creating new gene forms. Need to know, the 3 R's of gene physiology: genes can regulate themselves, replicate, and recombine to create new gene forms.
From genes to genesis.
If proving the existence of the gene solved the problem of heredity, scientists still sought to learn how an entire organism grows out of a single cell. Embryologist Ed Lewis of Caltech concluded that certain genes, effectors, start this process by turning other genes on or off. By experimenting with inchworms, which have a finite number of genes, Lewis discovered that each gene has a specific function to perform within the organism, with some even shutting off to cause its death. Genes, apart from being programmed with certain traits, were also affected by their proximity to, and interaction with, other genes. By way of analogy, the wood used to construct a ship contains certain intrinsic qualities, but it is the way the different sections are combined with the others that creates a floating vessel. Need to know, the way genes create a complex organism is determined not only by the traits they carry, but by their proximity to, and interaction with, other genes.
Part 3, The Dreams of Geneticists. Sequencing and Cloning of Genes, 1970 to 2001. Crossing over.
In 1973, biochemist Paul Berg created recombinant DNA for the first time by joining genetic material from different sources and using virus-bacteria hybrids as carriers. Shortly after, however, researchers Herb Boyer, Stanley Cohen, and Stan Falco discovered a way to create genetic hybrids with bacterial genes, which were far less hazardous than the virus-bacteria hybrids, and reproduce them in large quantities in an incubator.
This effectively ushered in the birth of a new world, in which genes could be manipulated for therapeutic ends.
Need to know, the creation of recombinant DNA and hybrids opened a world of possibilities for genetic manipulation.
The new music.
British biochemist Frederick Sanger learned how to read genes by copying a DNA sequence. Rather than break down the DNA into its component parts, he slowed its reproductive process so he could observe the order in which the parts appeared.
This brought science another step closer to altering genes.
For decades, scientists had managed to do so only by bombarding genes with X-rays and producing mutants. By the late 1970s, however, gene cloning and gene sequencing, determining the order of DNA nucleotides in a gene, were a reality. Need to know, the capacity to alter genes created a divide in the science of biology, between the old guard who merely sought to describe and classify organisms, and the new biologists who studied ways to change them.
Einstein's on the beach.
Scientists who gathered at Asilomar, California, in 1975 drew up a scheme to rank the risks of biohazards of experiments with recombinant DNA and restrict research accordingly. The conference espoused self-regulation by scientists in order to avoid having restrictions imposed later by governments. Need to know, the Asilomar conference recommended self-governance by scientists in gene experiments, but did not address the ethical dilemmas raised by the nascent science of genetic engineering. Clone or die. During the 1970s, genetic science began to move inexorably into the realm of technology and, by extension, into business. Researcher Herb Boyer and venture capitalist Robert Swanson formed the genetic engineering company, Genentech, which in 1978 manufactured insulin from recombinant DNA. Need to know, the founding of Genentech and its success in creating recombinant insulin marked the start of the biotechnology industry.
Up until then, insulin was still being obtained by a crude and inefficient method which consisted of crushing pig and cow pancreases.
Genentech patented the recombinant insulin and went on to invent many other genetic-based medicines.
Part 4, The Proper Study of Mankind is Man. Human Genetics, 1970 to 2005. The Miseries of My Father.
Late in life, Mukherjee's father fell from a rocking chair. This accident caused him to suffer from confusion, urinary incontinence, and difficulty in walking. He was diagnosed with normal pressure hydrocephalus, NPH, a condition thought to be genetic, although it requires external circumstances to trigger the symptoms. Need to know, the experience of Mukherjee's father illustrates how genetic science must address human illness, by considering genetic structure, its variants, the environment, and chance, all of which influence human pathology.
The birth of a clinic.
As genetic research in laboratories advanced, clinical studies were establishing the links between genetics and human illness. Victor McCusick founded the Moore Clinic in 1957 to study hereditary disorders. By 1998, he had discovered 12,000 gene variants linked to different medical disorders and traits. Need to know, The ability to detect genetic illnesses such as Down syndrome in fetuses created a medical industry in the 1970s, with the abortion of affected fetuses as one of its activities. Interfere.
Genetic testing on fetuses in the 1970s and the legalization of abortion was, in effect, a return to eugenics, which was now called neogenics, or newgenics.
The difference was that in newgenics, genes were used as the basis for selection by individual choice. Need to know, in the newgenics that arose in the 1970s, the genetic testing and medical procedures were carried out by individual choice, not mandated by governments.
A village of dancers, an atlas of moles.
By the 1970s, many diseases had been classified as genetic, but scientists were still unable to identify the specific genes that caused them. By compiling data on the affected families and using new mapping techniques, which pinpointed a gene's location through its linkage with other genes, researchers were able to identify the single gene that causes Huntington's disease. The same techniques were later used on cystic fibrosis. By the late 1990s, many genetic diseases could be detected through prenatal screening. Need to know, scientists' ability to map genes allowed them to detect genetic illnesses, opening a new horizon in man's ability to control human nature.
To get the genome.
The successful mapping of disease-provoking genes gave impetus to a much larger project, mapping the entire human genome, with its more than 3 billion base pairs of DNA. Estimates of the cost and the time it would take were huge, but so were the potential rewards in diagnosing diseases, particularly cancer. The Human Genome Project got underway in 1989. Need to know, starting in 1989, the Human Genome Project began to map the human genome, the entire encyclopedia of genetic information in the body.
The geographers.
Microbiologist Craig Venter decided to shortcut the process of mapping the human genome by examining only fragments of each gene. His company, Celera, entered into a race with the National Institutes of Health's Human Genome Project. In June 2000, the leaders of the two efforts announced jointly that they had made a first survey of the entire human genome. Need to know, in 2000, the Human Genome Project and Celera announced they had made a first survey of the more than 3 billion base pairs of DNA in the human genome.
The Book of Man, in 23 volumes.
This brief chapter summarizes the characteristics of the human genome in 23 bullet points and quotes, corresponding to the 23 pairs of chromosomes the genome contains. Perhaps the most singular characteristic of the human genome, compared to its animal or plant counterparts, is not the number of genes it contains, which is small compared to other organisms, but their complexity and diversity. The human genome has 20,687 genes, there are 32,000 in corn and more than 45,000 in wheat. However, the human gene network is marked by variety and by the sophisticated manner in which it operates. Need to know, the human genome, which is comprised of 23 pairs of chromosomes, is striking, not for the great number of genes it contains. Wheat, for instance, contains more, but rather for its complex and diverse nature.
Part 5, Through the Looking Glass.
The Genetics of Identity and Normalcy, 2001 to 2005.
So, we's the same.
The complete map of the human genome has revealed the similarities, rather than the differences, between races. The genes that determine racial differences comprise only a small part of our genetic makeup, about 7%.
The majority of genetic differences between people, in characteristics such as intelligence, personality, and temperament, occur within races, not between one race and another.
Genes themselves do not explain human diversity, that can only be understood by studying environment, culture, geography, and history. Need to know, the map of the human genome has shown that the majority of genetic differences exist between persons of the same race, not between one race and another.
The first derivative of identity. Genetics can determine some aspects of identity, but others depend on external factors.
However, sex, defined as maleness or femaleness, is completely genetic in origin. The presence of the Y chromosome determines maleness, but it must be activated by the SRY, sex-determining region Y, gene.
When this doesn't occur, confusion about sexual identity results.
During the 1970s, the idea that gender could be imposed externally, nurture can overcome nature, gave rise to experiments in sexual reassignment that completely ignored genetic factors, to tragic results. Need to know: genetics determine a person's sex as male or female, but gender identity is decided by a series of genetic and environmental factors.
The last mile.
For decades, science had taken the view that personality traits, temperament, and sexual preference were all the results of environment. However, studies with identical twins who were separated at birth and raised in totally different environments showed striking similarities in all these areas and more. For example, a pair of identical twin brothers raised apart in the Midwest both grew up to be chain smokers, stock car racing enthusiasts, and named their dogs Toy. The evidence, anecdotal and scientific, pointed to a genetic component of personality and gender identity. Need to know: genes can point to propensities in human behavior and personality, but they cannot determine outcomes, they cannot travel the last mile.
The Hunger Winter.
Survivors of the Hunger Winter of 1944 in Nazi-occupied Holland passed on the effects of that famine, heart disease, obesity, depression, anxiety, to their children and grandchildren. Research has shown that external events, whether traumatic or highly pleasurable, are recorded in cells by chemical markers that respond to various stimuli. The field of epigenetics studies changes in genes, which are inherited, but not caused, by changes in the DNA code. Need to know: epigenetics analyzes the permanent effects that external events, unrelated to changes in DNA, can have on genes.
Part 6, Post-Genome. The Genetics of Fate and Future, 2015. The future of the future.
Embryonic stem cells, are contained in an embryo and can reproduce every cell in a living organism.
They can also be extracted, genetically modified, and then reinserted into the organism.
This knowledge gave rise to the first attempts at gene therapy during the 1990s. One of the first patients to undergo such a procedure, 18-year-old Jesse Elsinger, died as a result. Gene therapy was put on hold for nearly a decade. Need to know: the early experiments in gene therapy during the 1990s were unsuccessful and in some cases had tragic results, putting a stop to such procedures for years. Genetic diagnosis, provivers. While gene therapy languished, progress continued in diagnosing genetic illnesses. Genetic diagnosis of ailments such as breast cancer or schizophrenia has created a new class of patients, called provivers, who know that they can be hit with a life-threatening disease and must decide how to use that knowledge. Need to know: the ability to detect the presence of genetic illnesses has meant that some patients now must live with the knowledge that they may be struck by a debilitating or life-threatening disease.
Genetic therapies, post-human.
Gene therapy comes in two distinct forms: alteration of a single gene to correct a deficiency, a change that will last only one generation, or permanent alteration of reproductive genes, called germline editing. A group of scientists in 2015 called for a moratorium on the use of gene alteration and gene editing technologies in a clinical setting, particularly in human embryo stem cells.
In a statement, they expressed concern over the possibility of initiating a slippery slope from disease-curing applications towards uses with less compelling or even troubling implications.
Meanwhile, Chinese scientists attempted unsuccessfully to genetically modify a human embryo. Chinese experimentation will continue, however, and the creation of the first human from a genetically altered embryo is not far off. Need to know: gene therapy can alter a single gene to correct a deficiency, or alter the reproductive genes, affecting a permanent change in subsequent generations of an organism.
Epilogue.
Our capacity to identify genetic illnesses and cure them has marked a huge advance in science, while at the same time posing a series of moral and ethical questions to the whole of society. According to the author, the influence of genes on humanity is richer, deeper, and more unnerving than we had imagined. Managing them, he says, will be the ultimate test of discernment for our species. Need to know: despite all the advances in human biology, including the mapping of the human genome, our knowledge of human genes remains very limited.